Um, and and today I'd like to talk about how some of our most commonly used drugs get into the brain. I'll do this by considering classic historic examples of drugs that have been or are currently used regularly and how they came into being. In most cases, I'll then pursue that and show how those drugs and their discoveries and understanding their mechanisms led to further discoveries of other classes of drugs as well. Here's some of the conditions and diseases we'll talk about today. Chronic pain, Parkinson's and Alzheimer's disease, headache, diarrhea, constipation, parasitic infections, seizures, obesity, CNS infections, viral infections in the brain, sedation, and brain tumors. So you see that we'll be talking in a very broad scope and a very broad area. And some of the concepts we'll consider besides blood-brain barrier will be the role of lipid solubility, transporters, acylation, particularly acetylation, caffeine, Trojan horse drug delivery, pharmacokinetics, pH partition hypothesis, absorptive transcytosis, the glycocalyx, lymphatics, focused ultrasound. Some of these are rather new topics in the field. Some of them are old topics. Some of them, I'll show you how they've transformed medicine, particularly the area of medicine that depends on drugs. So really, in some ways, this is a talk about the blood-brain barrier, that specialized capillary bed of the brain and how it's different, and therefore how drugs enter that tissue differently than they do peripheral tissues. This concept developed from work done about 1870 to 1920. And the most prominent figure in this period is Paul Ehrlich. Paul Ehrlich won the Nobel Prize 1908, essentially because he created the fields of pharmacology and pharmaceutical sciences. He's best known for his work on developing compound 664 and introducing the concept of a magic bullet, and developing arsenical based drugs that killed more spirochetes than people and therefore was considered a success. And Paul Ehrlich, when he was a student, in those days, if you got an MD degree and you wanted to become Herr Professor, you also needed to essentially get a PhD. And so his PhD work involved looking at the distribution of dyes in the body. Dyes gave rise to drugs. That's a whole story in itself, a fascinating one. But the point is that he used those dyes to develop drugs, and so he wanted to know where they go. So he injected this particular class of basic dyes into animals, and he saw that they distributed well throughout the body. You can see this is trypan blue, and you can see all the tail and the ears are dark blue. All the internal organs, peripheral tissues will be so blue you can't really tell what they are. But there was one exception, and that was the brain. Here's the inside of the skull, and there's not much here, but a little bit of dura mater. And yet it's taken up so much dye that you can barely distinguish what it is. And yet, less than a millimeter away is the brain, which shows no uptake of dye at all. So to jump ahead of the story, Paul Ehrlich got this wrong. He thought that this phenomenon occurred because the brain was incapable of absorbing dye. In other words, dye just wouldn't take to the brain. But it was about 1920 when one of his students, now a very famous professor, showed that brain tissue does take up dye, and therefore there was something blocking the dye from getting out of the bloodstream and into the brain tissue. But a parallel story we want to pick up is that of Felix Hoffmann. About the same time that Ehrlich was working on this, he joined the Bayer Company, and he had an incredible 2 weeks of work. On August 10, 1897, he acetylated a compound called sialic acid. Sialic acid was very commonly used to treat pain in this era. And I think we really don't realize how much pain was around at this period. We were well into the industrial area, and yet the common person, the blue-collar worker, was still most likely doing heavy manual labor. So we had pain from industrial accidents, and we also had pain from wear and tear of manual labor. And so people were really looking for ease of this pain. And this, of course, became aspirin. And this was a big advantage because salicylic acid had all of those side effects we kind of associate with aspirin, like, stomach upset and that kind of thing, but had it in spades. And it was also very difficult to use. To hit its therapeutic window. It was very short and chaotic, and so it really was a drug that was hard to use clinically. And the week after that, he diacetylated morphine. Morphine was widely used, and one of the problems with morphine was that it was addicting. And so he diacetylated this compound. It was marketed for cough and headache, again, by Bayer Company. And it was marketed as not being addictive. And it was very popular. Not only was it great at treating pain, but it also gave the user a confidence and a heroic feeling. And that inspired its name, called, which we call today, heroin. And it was marketed in the U.S. much longer than it was marketed in England as a non-addictive painkiller into the 20s or even into the 30s. And by that time, legislation and popular demand had realized it was a very addictive drug. But here we see a flyer from about the 1920s in America. Bayer Pharmaceutical Products, aspirin and heroin, and this is marketed out of New York. But it's still made by the Farben-Fabriken company. And this is a compound word. Germans love to compound words. Fabriken, of course, means factory. Farben's interesting. That means dye. So this is produced by the dye factory or the color factory. And this, again, goes back to the interesting history that pharmaceutical industry arose from the dye industry. Well, how to explain this? Why are these drugs so much better, the acetylation so much better than others, and where are they working and all of this? And this was sort of the ideas that people began asking post-20s. And over in this new area of cell biology, I'm not even quite sure cell biology was cell biology at this point, but it was getting there. Someone took some blue-green algae and looked at compounds. How do these compounds get inside a cell? And this is what they found. Here we have a measure of olive oil over water partition. And all of you cooks know that olive oil is oily and water is watery or aqueous. And so the partition coefficient, you stick this in, you shake it up, shake the drug up with this compound. The two phases separate and you measure what's in each phase. And therefore you can give a scale of how lipid soluble or how water soluble something is. So for example, 0.001 means that 1 out of 1,000 molecules enters the olive phase and that kind of thing. You'll see, I'll go to this concept a lot, and you'll see these numbers change because different phases of lipid or oil was used in different studies, and the partition coefficients shift, but the concept stays the same, that if you are lipid soluble, you get into cells much more easily than you do if you're water soluble. And the second criterion that was a little bit more, it was a little bit important, was the size. So small lipid soluble molecules get in much better than big water soluble molecules. And this became a tenet in cell biology. About 1940s to 1950s, when this man was a young guy, mid-career, he made two great contributions to today's story. This is Hugh Davson, and he developed about 1930s, 1940s, the Davson-Danieli model of the cell membrane, which we'll talk about in a moment. And he also showed experiments showing that the blood-brain barrier, this barrier function, resided at the level of the capillary. And Hugh Davson did so many things. He was sort of a classic English polymath. And I had the pleasure of meeting him the first time I went to a meeting in Europe. There was this rather elderly man at 10:30 in the morning. I think he also was tired of the science meeting. drinking schnapps there, and I have no idea who he was. Had a great conversation, and he was a very brilliant guy. And here's a sort of a souped up version of the Davson-Danieli model. It's also called the bilipid model. And basically what we have, I'm understanding, you can see my pointer. So you have these two lipid layers that come together. And so in this way, you present essentially a lipid soluble surface. And so this explains why things can just sort of meld in to the membrane and get across. Later, this was enhanced by realizing there were proteins embedded in the cell membrane, and there all this sticky, gooey stuff, which May Reed tells me many people call it brain snot, although it's on any cell surface, the glycocalyx or the basement membrane, depending on what your orientation is. So he contributed that, and that began to explain how lipid soluble things could get in. That became sort of a dominant theory. But his other idea that he published about the 40s and 50s, that the blood-brain barrier was residing in the capillary of the brain, ran into some major difficulties. The first one being that if you look at the blood vessels that course through the brain, they don't look any different than those that course through the rest of the body. So how could this have distinct features? My field today loves to say that the brain is highly vascularized, and that if you take all the capillaries in the human brain, you string them out, they'll reach 600 miles. I'm not sure why you would want to do that, but it's an interesting statistic. But here you see that the capillary density is not particularly greater than that of any other organ. It certainly doesn't compete with the density of, say, the liver or the kidney. So, in short, gross anatomy doesn't tell us or support the idea of a blood-brain barrier being there. Nor does microscopy. Here's a capillary bed from the brain, a capillary from the brain, and it doesn't look any different than capillary from any place else. So in some ways, Davson's theory was really in trouble. And then in the 1960s, late 1960s, a couple of guys, particularly a guy named Rees, along with collaborators like Karnofsky and Brightman, turned the electron microscope on to capillary beds. And there we got the answer. So for comparison, here's the liver. The liver is one of the most leaky capillary beds in the body. And of course, leaky capillary beds are essential to the survival of the tissue, because that leak is the ultrafiltrate that nourishes all these tissues. And here we see there's a bunch of holes punched in here. And those are microvesicles, or vesicles, macropinocytosis, micropinocytosis, all those guys crossing across the layer, the capillary bed, the capillary wall, some people call it, nourishing the tissues. And then there's all these little funny looking holes. They almost, to me, look like cathedral windows. And indeed, they're called fenestrae, the Latin term for windows. And stuff is going through there. And then you can see the edge is rather ragged, and sure enough, where capillaries come together in the liver, they don't really fit very well. So there's leakage between the cells as well as across the cells. Those three things are all pretty much gone in the blood-brain barrier. Where cells come together, they're cemented by these tight junctions. There's very little pinocytosis, and there's no fenestrae. And so there's essentially the brain presents to the given drug a solid membrane, lipid-soluble membrane. And then Oldendorf actually measured brain uptake as a function of lipid solubility. Here's an early study that morphine doesn't get into the brain very well. You put one acetyl compound, you get codeine, gets in better. Put them in two, you get heroin. And so these substances are really crossing the blood-brain barrier well. And so we can begin to see how these can just meld across. And perhaps a better illustration of this will come up soon. But then Oldendorf went one step further, and he produced this rather monumental paper where he looked at all these common compounds against their lipid solubility versus brain uptake method. And now we can see that morphine is not very permeable. It gets in, but not great. But heroin, which is about 30 times more lipid soluble, gets in about 30 times better. And eventually this all levels off. But so Harrison produced this great cartoon, which I really like. And it just, to me, sort of summarizes, this is how we think, we sort of envision drugs getting through the lipid soluble membrane. So the characteristics of the transmembrane diffusion, as it became called, are non-satchable. So the more you get into the blood, the higher that you make the blood levels, the more that will get into that, not a satchable system. And it is mostly dictated by lipid solubility, although molecular weight, square root molecular weight can be used as a correction factor, because the bigger the molecular weight, that's sort of a drag. And some of our favorite drugs and most important drugs, besides morphine, ethanol gets in this way, and nicotine gets in this way. And so you think that everything would be solved. Now we can just make small lipid-soluble drugs and get them into the brain, and we can treat anything we want. But life is never that simple. And for the first problem, you can have too much of a good thing. And sure enough, you can have too much lipid solubility. First, if you're lipid soluble, you'll not only be able to cross the blood-brain barrier better, you'll be able to cross all other tissues better too, including, for example, the liver. So you're always doing this dance to make it more lipid soluble so that you can enter the brain if your brain is the target. But if you make it too lipid soluble, taken up by all the other tissues, blood levels decrease, and although you may have a higher transfer rate, you'll have less material to transfer. So it's a dance here. Another problem is you can get caught in the membrane. I'll talk about that a little bit more. You can become a PGP substrate. We'll talk a lot more about that in a different section. Or you can become too powerful. Morphine versus heroin is sort of an example of that. And indeed, that's been repeated. There's this paper in Nature in '77 by Gayden and Romer. He fluorinated some peptides, opiate peptides, and they're so powerful that they couldn't really be used clinically. And fluorination is one way to make something more lipid soluble. So again, you can get stuck in the membrane. So think about it. Imagine you're a little molecule and you're going down the bloodstream, and you say, "Oh, I want to get into the brain." And so there's a lipid layer right there. So this is what you've got to do. You've got to get out of the blood and get into the lipid layer, right? So therefore, you need to be lipid soluble. But then you have to transfer back into the cytoplasm, which is an aqueous media, and then back into lipid solubility, and finally back into the aqueous media of the CSF or interstitial fluid. Now, some people alter this model. There's always been this idea, it's been hard to show, that once you're in this membrane, you just truck on around here behind the tight junctions and come out on this side. And in that case, you still have to go aqueous, lipid, aqueous. So the best molecules are a little bit of both. And here's the paper by Stachino. Here he's used octanol as the oil, and this is log. So here's one. So 1 is, of course, 10, and 0 is 1, which 1 would mean that you distribute equally into the lipid and into the aqueous phase. And you see that somewhere between 1 and 10, you sort of peak out, the maximum extraction you can get. And after that, going up more doesn't help because you're sort of now stuck as much as is getting in. And if you keep going, you actually get less material penetrating all the way through and getting into the brain. And that's because you just get here and you love it so much you don't want to go back into that wet, cruel world of the aqueous environment. So lipid solubility clearly is an idea that's been around since the early 20th century, and it's been revised. And it's been best revised by Lipinski, who has his rule of 5. And what you might first notice, interestingly, about the rule of 5 is there's only 4 rules. And I think what Lipinski meant, all those 4 rules have 5 involved. So, for example, 5, 10, 500, and 5 here. And I think what he meant, rule, is the way an emperor rules, the domain, the influence of the 5. And this was sort of an early 21st century upgrade of the Lipit hypothesis, sort of broke it down a little bit. So you're not likely to cross if you have more than 5 hydrogen bond donors or 10 hydrogen bond acceptors. And this is a play on lipid solubility. And if your molecular weight is greater than 500, you're going to have trouble. That's, of course, the molecular weight component. And then if your calculated log PI is greater than 5. In other words, if you're too lipid soluble, you're going to have trouble. And so this, what Lipinski did was he works for an industrial company, pharmaceutical company, and he took all of their compounds that they were developing, I think 20,000, picked use criterion, got down to about 2,000, and they had looked at their ability to cross this monolayer of cells. The Keiko-2 cell would derive from a colon cancer, and was able to show that these were the principles that best dictated those drugs got in the best. But right away, he noticed there was a BRO5, and this is the way it's referred to in the literature, BRO5, beyond the rule of 5. These were compounds that did not follow the rule of 5. Antibiotics, antifungals, a lot of vitamins, cardiac glycosides, of course, drugs that use transporters, microcycles maybe, and peptides. He excluded peptides from his calculations. I'm not quite sure why he did that, if he did it retrospectively because they didn't fit, or because he realized they were just structurally different than all those small molecules. But at any rate, that was kicked out. And if it's work we did way back in the 80s, when we were looking at the ability of peptides to cross the blood-brain barrier, and we purchased every peptide that was then currently available that was radioactively labeled, and looked at the ability to cross the blood-brain barrier. And these were an exception a little bit because they're a different class of drugs, or largely opiate peptides, and they play by a different game. But most of the others more or less follow lipid solubility as a rough estimate. And we also looked at a series of DSIP analogs. DSIP is delta sleep-inducing peptide, and this was derived from the cerebrospinal fluid of sleeping cats by Monnier and Schoenberg. I do not know to this day how you get CSF out of a cat who's asleep. It seems they would wake up, but these guys were Swiss, and Swiss scientists can do all kinds of miraculous things. And then they showed that if you take the same compound, you stick it into the brain of an awake cat, the cat goes to sleep. So a really powerful substance. There's a lot of interest in this soon after they published it. And this was an era there's a lot of interest in developing peptides as drugs, and a lot of interest, for example, in opiate peptides, because it was believed you could take an opiate peptide, if you could just get it to work, just get it into the brain, it would be non-addicting. And to jump ahead of that story, they've created peptides that could cross and deliver analgesic properties, but they were still addicting. But here we have a series of DSIP analogs, and we see that they do indeed cross at different rates. And that here we have the calculation predicting how well they cross. And the major thing here is the lipid solubility. These two are the pharmacokinetics, which just measures how well they stick around in blood. Again, you've got to stay in blood to reach the blood-brain barrier in order to cross it. And these just say, you know, this is correction for things that are cleared by the blood faster or slower. So the main characteristic of these drugs is optimum. But unfortunately, peptides tended to be very hard to harness pharmacologically. And so the interest drifted away from the 90s and 80s and 90s, it waned. And then the beginning of this century, peptides came back in a whole different way. And here's some compounds I know you know, the incretin receptor agonists. and their use in the treatment of insulin-resistant diabetes. So just to recap this, the incretin effect, which is key to the idea here, is that if you give glucose intravenously into the bloodstream so that you produce this curve, and you compare that to glucose you give per aura so that the blood glucose level is identical, and now you look at what happens to insulin release, it is not the same. If you give the glucose orally, you get a big bolus of insulin release. If you give it directly, intravenously, hardly any incretin effect at all. And the reason is because when the glucose is absorbed across the GI tract, across the gut, and into the blood, it stimulates the release of GLP-1 and also its sidekick, GIP, glucagon-like peptide. And that, in turn, influences the release of insulin from the pancreas. So this is a mechanism by which the body helps to control postprandial glucose levels. And it's become, of course, a centerpiece in the treatment of diabetes, as has another class of drugs, DPP-4s, which essentially are enzymes that prevent the cleavage of GLP-1. So because the incretin effect is not working in type 2 diabetics. So this is a play on reintroducing the incretin effect. And I know that we all have heard this other life it's got now, which is as a dietary measure, Ozempic. So what's Ozempic? Ozempic is also known as semaglutide. It is an analog of GLP. It's truncated, only 7 amino acids, 7 to 37. And guess what? It's acylated, but it's acylated in a different way. Instead of acetic acid, it's got this very long, bodacious, free fatty acid chain. And this does a bunch of things. It helps to, now we're beginning to understand at this area, what does acylation do? When it's acetic acid, it often helps you, all acetylation increases lipid solubility. And the other thing it tends to do is to help tightly bind to albumin. Remember, salicylic acid didn't work too well because you had to give high doses and then they cleared very quickly from the blood. So in that case, acetylation of salicylic acid helps it to stick to albumin, longer half-life, more even therapeutic effect. So here's semaglutide, as opposed to another GLP analog, Exendin, which usually has to be given twice a day. Semaglutide, because it sticks around, give a higher dose, but you only give it once a week, that kind of thing. And of course, there's lots of effects that these things do, as you can imagine now, but they also work in the brain. And so they have been shown to have a lot of effects. Epidemiologically, they look protective in AD, as well as Parkinson's disease, maybe even stroke. And in neuroscience studies, they show evidence of neuroprotection, neurogenesis, proliferation, and differentiation. So now the big question is, how do they do this? Well, of course, if they protect against diabetes, they're going to reduce the risk of stroke and various and all the macrovascular and microvascular damages that are associated with the diabetic drugs. I'm sorry, with diabetes. But here's the thing: they work in the brain. They seem to work in the brain directly and rather rapidly. And so the idea is, are they working in the brain independently of what's going on over in the body? And spoiler alert, the brain does have GLP-1 receptors and GIP too. So the question becomes, can they cross the blood-brain barrier? Can they get into the brain to hit those GLP receptors? And the answer is sometimes. So here's a combination of two studies that we did with collaborator Conrad Talbot, who was the first guy to show that the Alzheimer's disease brain, human brain, has insulin resistance, CNS insulin resistance. He was able to get brain autopsy tissue fresh enough that he was still able to measure the function of insulin receptors in hippocampal tissue. And so Elizabeth Rea and I were fortunate enough to work with him, and we've tested 13 different incretins, all the ones that are on the market, as well as some that are in development and or just developed because they're whatever. And here's what we found, that a lot of them cross, including our old friend exenatide, and 4 of them do not. And those 4 are all acylated. So in this case, acylation is preventing transport, and maybe that's because they're binding to albumin so tightly that they can't get in. There's no free drug. So how do the rest of them get in? And that, believe it or not, we've just figured out, and we just figured it out this week. We had published another mechanism, realized that the company had sold us a different kind of peptide than we thought we had. So when we did the recalculations, we found that the remaining 9 substances cross by a function of their lipid solubility. And if we correct from that molecular weight, we get even more. So we have completed this cycle in the 21st century that started out in the late 19th century, showing a major class of peptides, how they're affected. Acylation inhibits passage, lipid solubility enhances. So one more concept before we leave this idea of how this all sort of started back with Hoffman. And just to recap what we said so far. So morphine doesn't get in too well, water soluble, and heroin gets in very well because it's lipid soluble. But heroin is not active. Heroin doesn't plug in to the opiate receptor. Heroin doesn't have an effect. And so this brings us to the concept of latentiation, or as they say nowadays, the more popular term is prodrug. And what happens is once heroin has gotten in, acylases deacetylate it and change it back into morphine. And now morphine is trapped in the brain because just like water-soluble morphine can't cross lipid barriers in one direction, they can't cross the lipid barriers in the other, so it becomes trapped in there. So heroin is a prodrug. It's a delivery mechanism by which it becomes a drug after it reaches site of action. So all those stories you can see sort of developed from early 19th century work. And now I want to give much briefer synopses of other ideas that have worked. And this one is one of my favorites. It talks about brain-to-blood efflux systems, and we'll go from everything from diarrhea to dementia on these. And these are all the others are going to be much shorter than the one I just did. So the idea here is that we have transport systems, but they're directed from the brain to the blood, and we have 2 big categories of that. Those where the transport system is here on the brain side of the cell membrane, and maybe a companion one here. And so they sort of vacuum things out of the brain and across the capillary and into the bloodstream. And then we have LRP1 was an example. And then we have this one, P-glycoprotein, where it's stationed in the blood side of the membrane. And what it usually does is when xenobiotics try to cross, it grabs them and spits them back out. So they never get a chance to get into the brain. And PGP is, again, probably the most clinically relevant transporter in the body that no one's really heard about it. It's also called the ABCB1 transporter, or the MDR1. Cancer cells, when they become resistant to their chemotherapeutic, it's because usually they've developed this transporter that spits the chemotherapeutic back out. It's also in the gut and many other tissues where, particularly in the gut, it guards against bad things coming in from the diet. And in the brain, it protects against xenobiotics. And so many of you may have children or grandchildren, and they get diarrhea. And so you could treat them with morphine because morphine induces constipation, opiate-induced constipation. But unfortunately, it results in an obtunded baby. And for some reason, many parents don't like obtunded babies. So instead, we treat them with drugs like Lomotil and Imodium, and on our older patients too sometimes. And that they're effective because they have oxalate or loperamide, which are opiates, but they are strongly binding to PGP. So the PGP keeps them out of the brain. So you have a happy baby. Both ends are working well. But if you have a knockout animal, PGP, and you give these drugs, they get into the brain very well, and they're have the same set of CNS effects as morphine. So there's lots of PGP substrates. It's a very ubiquitous, very promiscuous transporter, and it has more classes of drugs than most transporters have actual drugs. So opiates, protease inhibitors, calcium channel blockers, steroids, antibiotics. We talked about anti-cancer drugs already and how that's important to treatment of cancer. Cardiac drugs, a whole bunch of odd things like phenytoin and colchicine and some of the lipid-lowering drugs. And the basic idea to think about this is if, and some of these are very weak binders to PGP, like morphine is pretty weak, so it still gets in enough to have an effect. Others are very strong, like loperamide, and therefore it doesn't enter. And one way to think about it is if you're a very strong effluxer, then you can use this drug and it doesn't have CNS effects. And if it isn't the PGP substrate or it's a weak one, then it gets into the brain and it may be able to treat diseases. So that is sort of the dichotomy of this area. Mother Nature is full of efflux systems, and we are really discovering them. I think this is one area that really promises to add a lot to our knowledge of not only medicine, but life in general. So here's my favorite example of an efflux system. How can the tobacco worm eat the tobacco plant? Because nicotine is toxic, and the answer is because it's got a efflux system in its little hemolymph to keep the nicotine out of its ganglion. And so that's how it works. And I know many of you are raised tomatoes and that kind of thing, and you're going to say, this looks like a tomato worm to me. In fact, it's on a tomato plant. And yes, it's true that Manduca genus loves solanaceae, so they will eat anything, but they are still rather specialized. You know, the tomato worm prefers tomato. Nicotine, the tobacco worm can actually eat tobacco plants. So I thought it would be fun to educate you about the difference between these two worms. That the tobacco worm has 7 stripes that have no border. I know it looks a little black, but they say no border, and its horn is red. Whereas the tomato worm has these 8 V stripes, and its horn is black. And the mnemonic for this is this looks like a cigarette, hence tobacco, with the flame of the cigarette, whereas the V stripes are vine ripe tomatoes. So there you go. PGP substrates are involved in all kinds of clinical aspects. One of the big ones are in seizures. Almost every drug that has anti-seizure activity is a PGP substrate, with the exception of valproic acid and anakinra. Anakinra is an interleukin-1 receptor antagonist agonist. About 30% of the population overexpress PGP, therefore less drug in the brain. About 25% underexpress, therefore more drug in the brain. And this may explain why about 30% of epileptics are rather resistant to most seizure medications, with the exception of valproic acid. The other thing is that PGP not only shows genetic variation, and the activity is altered by a series of physiological events, such as inflammation. And when a person has seizure activity, neuroinflammation goes up dramatically. And therefore, this may explain why status epilepticus is so hard to treat, because the more seizure activity you get, the more inflammation you get. The more inflammation you get, the less well the better PGP activity works. And so you are pumping out the very drugs that you need. Supporting this is a series of infantile seizure disorders that are induced by a prodrome of inflammation, like a viral infection or something like this. And these kids just start seizing, hundreds of seizures a day, resistant to all the anti-seizure medications, with the exception of anakinra, a non-PGP substrate. Other clinically relevant efflux systems, in Alzheimer's, in you and I, hopefully, we have good, healthy PGP and LRP1 systems. This is pumping out the amyloid beta from our brain so it doesn't, accumulate and form plaques. Both of these transporters are lost in Alzheimer's disease. Indeed, PGP is lost in every animal model of Alzheimer's disease as well. Metenkephalin is involved in, which is an endogenous opiate. Not all opiate peptides have the same portfolio, and this one seems to have a lot of anti-seizure activity. It can be used if you have a seizing animal who's seizing because they've been withdrawn from alcohol. You inject enkephalin in the brain, they stop seizing. Likewise, it seems to be that alcohol suppresses the production of metenkephalin and therefore may make them more susceptible to seizures. There's a transport system, an efflux system, PTS1, gets rid of metenkephalin from the brain, and that is also suppressed when animals are alcoholic. So that's good news. Make less metenkephalin, but you're pumping it out less fast. The problem is when the animal stops drinking, it takes about a week for metenkephalin expression to come back online, but PTS1 recovers in a matter of hours, pumping out the residual metenkephalin, and perhaps that's the basis, one of the bases of alcohol withdrawal seizures. Many anti-HIV drugs are effluxed, and they don't really reach therapeutic levels because of the efflux system. There's a brain-to-blood transport system for IgG, and of course, many of the drugs we're trying to use to treat Alzheimer's, like lecanemab, are IgG molecules, and therefore are not reaching the therapeutic levels they would. They're about half what they would be as if there were no efflux system. And the PGP decreases with both age and dramatically so with AD. Therefore, more drugs, those substrates are getting into the brain. Maybe this explains in part why Alzheimer's disease patients and aging patients and Parkinson's patients are more susceptible to some drug toxicities. And then there's some evidence that PGP may be involved in delirium. Running a little long, so I think I will skip that. The Manuka beans and Trojan horses are an interesting play. About 600 BC, the Ayurveda literature described a condition that sounded like Parkinson's disease and noted that it could be treated by ingesting macana beans. And about 1913, Guggenheim isolated L-Dopa from a very similar plant. And in 1911, Funk was beginning to do investigations of L-Dopa, and he was giving it to himself and decided it was too toxic to use in patients. And this stopped the use of L-Dopa for decades until Coetzee and Yahr in 69 and 67 reintroduced L-Dopa for the treatment of Parkinson's. And so how does this work? Because how do you treat Parkinson's? Dopamine is very water soluble, doesn't cross the blood-brain barrier by that mechanism. The dopamine transporter isn't in the blood-brain barrier, so you can't use dopamine to treat Parkinson's for this reason. It doesn't get into the brain. And Oldendorf again came to the rescue. He discovered that the large neutral amino acids, you see that they get into the brain much better than the lipid solubility predicts, show that they're crossing by a satural transport system. And so here's to remind you the dopamine biosynthesis pattern. L-phenylalanine is converted to tyrosine, tyrosine is converted to L-Dopa, and then that's finally converted to dopamine. So all three of these are transported by the large neutral amino acid transporter, or LAT1. LAT1 is expressed about 100 times more in the blood-brain barrier than in other tissues, and therefore you sort of have a specific brain delivery system for these. And originally it's interesting, but they all compete with each other. That's a very fundamental point to remember, that because they're all being transported by this, if one goes up, it will sort of block the entry of the others. And so this explains perhaps why Cortias was unsuccessful in treating Parkinson's by using L-phenylalanine. The reason he did this, because L-phenylalanine is water soluble in comparison to tyrosine, which is very lipid soluble. So you can't get it into an aqueous solution, so you can't give it to patients. But phenylalanine is relatively water soluble. But the Parkinson's patients actually got worse. And the fact that there's competition probably explains that. By giving too much phenylalanine, he blocked tyrosine from getting in. And so therefore, less dopa was actually in the brain. This principle is used to treat phenylketonuria and maple syrup urine disease, the latter being too much of the branched chain amino acids. Tryptophan has been used to treat depression, and leucine blocks kynurenine, and kynurenine is thought to be responsible for the inflammation-related depression. So inflammation will increase this enzyme called IDO, which increases tryptophan to kynurenine transport conversion. And kynurenine crosses that LAT and gets into the brain and then causes all kinds of terrible things. And so how could you block this? And one possibility is leucine. It's fairly water soluble, can get in good levels of leucine into the bloodstream. And sure enough, in preclinical studies, leucine blocked kynurenine and improved depression. And now there's a clinical trial for that, leucine in midlife depression. So there's lots of drugs that there's lots of plays on this kind of thing with the large neutral amino acids and classic drugs, including gabapentin, get into the brain this way. But about 90–95% of the drugs we use for brain diseases are small lipid compounds, and not many drugs have been developed to use transporters. And so eventually, I think this will become a major area of transport of drug delivery to brain. Caffeine, for example, is one of my favorite examples of things getting in. I'm running a little behind, and I want to leave some time for questions. So I'm going to talk about one other thing that I think is really cool. And it's kind of one of the latest things. And there have been decades and decades of trying to disrupt the blood-brain barrier to allow drugs to get in. And originally, it started out about 1884 when Rickman Godley performed the first successful resection of a primary brain tumor. And he and Bennett figured out where that tumor was in the brain by doing physical exam. But it was a classic example of this. The surgery was a success, but the patient died about 4 weeks later from infection. So people realized at this point, maybe it'd be better to treat things with drugs. And here are some principles that we have to worry about with brain tumors. The main one, though, is although brain tumors are more leaky than the blood-brain barrier, they're still not. They're still very, very tight compared to peripheral tissues. And so there's always been this interest in how do you disrupt the blood-brain barrier enough to let your drug in and not to let everything else in. One of the latest ones is focused ultrasound. And I got to admit, I'm a big critic of this because if you disrupt the blood-brain barrier, you let everything in of that molecular size. And so it usually winds up not a pretty picture in terms of neurotoxicity. But here's a study by Razak, just published, in which he used ultrasound to deliver more aducanumab in an Alzheimer's disease brain. So here's the background. Here's a baseline brain, and he's going to introduce some focused ultrasound to this region of the brain. And then after doing that, he used a gadolinium to show, yes, look, there's all this leaky area in the brain. And here's a close-up if you can't see that. And then about 48 hours later, all the gadolinium is gone. So this is great. I can disrupt the blood-brain barrier willfully in a reversible way. So now he took 3 patients and he gave focused ultrasound to a part of the brain, a small part of the brain, just like he did here, and not, of course, to the rest of the brain. And here's what he found. The more red is the more A beta. So, of course, aducanumab is supposed to clear A beta from the brain. So here in this patient number 1, see how much red there is, and then 26 weeks later, a lot more blue, a lot less red. A lot of red, mostly blue, and it goes on down the line. We'll pick another one. Here's one. A lot of red here, a lot of blue. So he's saying focused ultrasound is letting more antibody in, which is clearing more drug. And he sort of did these, every treatment, he would sort of measure how much aducanumab was getting in to one side of the brain. And I'm sorry, how much was being cleared on the non-FUS side of the brain versus the FUS side of the brain. You see it looks rather dramatic. So perhaps this is a new way. He's talking about doing a big clinical study where he does focused ultrasound on all of the brain, which would be really, really tough to do and probably take a long time. But hey, you've got 2 weeks between each lecanemab dosage to do that. So that's sort of my talk. We talked about classic lipid solubility and efflux systems, a little bit about transporters. I talked too much and didn't talk about charge. Maybe we can do that some other time in blood-brain barrier disruption. And here is your QR code. If you haven't used this before, you just point your phone. When the little bar comes up, hit it, and then you get all this evaluation. Just put top ratings for everything, and then the little space say, the sky is great, you should get a raise. And that's how you use that. So with that, I'm happy to take questions. If anyone's still awake.
Everyone is wide awake because we have our caffeine that is getting into our brain. They taught us all
Right.
about. If anyone has questions, feel free to unmute. Amy Thomas has a question in the chat. Are you able to see that?
I'm not very fast with chat, I'm afraid. There it is.
Oh, she wants part 2.
Good old heroin, yes. The drug that keeps on taking.